Okay, number 2. So let's see. Now we got what the skin is for. Let's take a look at its multiple layers. We've already talked about the epidermis portion and the dermis portion. So here we're now getting even closer to the epidermis to look at its multiple strata. Right? Each strata is a layer of different cells, and so a single one is a stratum. Right? And so most of it is made up of a type of cell called a keratinocyte, a cell that is meant to generate keratin. Right? And let's see. The cell life cycle begins all the way down here at its deepest layer. This layer is called the stratum basale, like base or basement. Right? And in the stratum basale, you have about 4 different types of cells to be found here. And the first one are these, stem cells. The job of any stem cell is to eventually differentiate into other cell types. Now, what this cell is mostly going to be turning into as it divides over and over again are keratinocytes, the ones that eventually grow to the surface and get flaked off. But there are a few other cells that could form from this stem cell. Right? The majority of the epidermal cell of the keratinocytes will synthesize keratin, so that's most of these pink ones. But you also got these, melanocytes. Melanocytes are cells that are going to be producing the pigment melanin, so that's why this one has been given sort of a darker brown color. Right? Melanin is a protein. You can assume that, not someone assume, you can make that judgment based on the fact that it ends with IN. Right? So this protein gets synthesized by the melanocyte, and then it gets packaged into little discrete vesicles, those are all the little dots here, and then they get exocytosed into the surrounding tissue. So all the keratinocytes that are nearby the melanocyte will now absorb the pigment of melanin that the melanocyte released. And then that pigment gets taken in, and it gets collected over the nucleus of each of the cells. It doesn't necessarily spread evenly throughout the cell's cytoplasm. It almost makes an umbrella over the nucleus of every keratinocyte that gets melanin. Why there? Well, the melanin's job is to absorb ultraviolet radiation so that it can't penetrate deeper. And what would be just underneath that melanin would be the DNA of the living cell. So consequently, we now protect specifically those important instruction molecules for the cells that are doing a lot of the reproducing. Okay? But this means that the melanin made by one cell can only spread so far, which is why you should find numerous melanocytes all over the stratum basale of the skin in, say, a person who has an even skin tone. Now, I'm a freckly individual, so any freckles on the back of my hand show the irregular distribution of my melanocytes. I would have a cluster of them, I don't know how many exactly, but just a cluster of them in a small patch of skin, and so the cells above those melanocytes would have melanin in them, making the darker freckle color. But in between the freckles, I'd have large areas where there are no melanocytes, and so the skin appears paler. Interesting thing about this is that if you have a person with darker skin, they don't necessarily have more melanocytes. They will probably have a more even distribution of them, but the quantity that of melanin that the melanocyte is producing, that's higher. So same number of cells, just a greater level of production, and even the melanin can be a bit different. The melanin of a darker skinned individual doesn't degrade as fast as in a lighter skinned individual. So I don't have an explanation as to what the difference is between those two types of melanin, but one more thing about the melanin though. It's only going to be prevalent in the living cells of your skin. Once it gets up here to where the cells die, then the melanin degrades because it's exposed to light, it's exposed to a different pH, different temperature, so it's going to go through some denaturation and can't do the job that it did before. Which is why you may note that on the palms and soles, where you have thick skin, lots of dead cells, of people with darker color skin, that those areas appear lighter. That's because there's not as much melanin visible through the extra layers of non-melanized dead cells. Okay? Alright, but still, melanocytes are one of the other cell types in the stratum basale besides the stem cells and the keratinocytes. And then we've got this one, a tactile cell, or Merkel cell. Merkel for the guy who described them first, because anatomists like to name things after themselves. But tactile cells give you a tactile sensation, which means it's a sense of touch. This is not the only cell that's involved in giving you a sense of touch, but it's one of your receptor cells. The tactile cells are epithelial, though. They're not neurons. However, underneath every tactile cell, there's going to be a thing called a tactile disc. This little yellow blob right here, that is the dendritic end of a neuron. So when you press down onto the skin in a very light fashion, like an ant walking on the surface of your skin, then that impression is going to push on this cell and create a deformation of its membrane. The cell will, as a result, release a chemical onto the surface of this neuron. The neuron will pick up the chemical, and the neuron will now create an electrical signal that is sent off to the brain. Right? So without the Merkel disc, the neuron would not be stimulated, because that's the only cell that's going to dump this chemical onto the neuron surface. And now, mind you, there will be other nerve endings that are stuck in the dermal portions of the skin and even the epidermal portions, but this one gives us a sense of light touch and texture, your Merkel cells, or tactile cells. So it's part of your sense of touch. But those are your four major cell types that you will find inside the stratum basale alone. Stem cells to eventually become all of them, keratinocytes, the most common cell type, melanocytes to make melanin, and then tactile cells to give us a sense of touch, part of our sense of touch. Alright, now as we go upwards, once you get away from the bottommost layer of cells, then you are now in the next section called the stratum spinosum. The reason it's called spinosum is because it appears that there's tiny little spines of cytoplasm that are extending between one cell and the next. This is an affectation of making slides. Because when you make a slide, you have to take a very thin slice of skin and put it on the slide and then pour chemicals on so things get stained. Well, almost inevitably, some of the cells are going to dehydrate as you make the slide. And when it dehydrates, these cells start to pull away from one another. But the places where they are still kind of firmly connected to one another are places where you'd find desmosomes, like little buttons that are holding two cell membranes together, and that's where you see all the little spines of the stratum spinosum. Okay? But nonetheless, once you get away from the stratum basale, the next layer of cells up is stratum spinosum, and then it stays stratum spinosum until you get to these dark speckled cells of what's called the stratum granulosum. Okay, so pretty thick stratum spinosum, and is dominated primarily by keratinocytes. There may be a few of them that are still reproducing, but they're going to be in the deeper layers. Once you get about 3 or 4 layers of cells away from these blood vessels, they stop reproducing. But here's another inhabitant, a dendritic cell, also known as a Langerhans cell. Dendritic cells kind of look stellate, and so they look like neurons, but it's packed in here with a whole bunch of epithelial cells. These dendritic cells are wandering macrophages that live in your skin. So that say that there is some sort of pathogen that manages to make it down through the dead layers, well, now you've got this population of patrolling cells that are going to be looking for things that don't belong. And if they get hold of them, they're going to eat them. Okay? So you've got this set of protective cells patrolling through your skin constantly. Okay? So those are your dendritic cells, skin macrophages. They typically stay just where the living cells are, although sometimes they can also migrate down here into the dermis sections as well. Okay? So let's see. Stratum basale, 4 cell types. Stratum spinosum, really just 2 cell types, the keratinocytes and the dendritic cells. And then we get to the next one, the stratum granulosum. All right. Stratum granulosum. Consists of 3 to 5 layers of flat keratinocytes, so they're becoming more squamous as they're getting pushed to the surface. More in thick skin than thin skin, and what makes them granular are the fact that they have these dark staining vesicles inside of them. In those vesicles is where you're going to find a waterproofing keratohyalin material. Right? It's a glycolipid, right? And this glycolipid will eventually be secreted outside of the cells. Right? In fact, it will be one of the last things that these cells do before they die. Right? But now that we've got this glycolipid in between the cells, it helps to create more of a waterproofing layer between the cells, so it's harder for water to seep in between them. And then your stratum granulosum cells, as they get further pushed toward the surface, die because they're so far away from the blood. And those dying cells tend to make this indistinct layer that's called the stratum lucidum. Now, stratum lucidum is pretty hard to identify in a thin skin section. So let me just jump back to one or two of those. Right? Let's see. Okay, here's a good thin skin area. And there are your dead cells on the top, and there where it's darker purple, that's where your living cells are. So between there and there, it's kind of hard to identify a stratum lucidum. Right? This one here as well. Right? That dark layer of cells are part of the stratum granulosum, and then these are the dead cells, kind of already starting to flake off on the top. So stratum lucidum is a little bit more difficult to identify. In thick skin, it's generally easier. Right? Here are your living cells from here to here. This is all dead tissue, and there, that slightly clear area, that's your stratum lucidum. It's pretty thin. But stratum lucidum you should mostly assume that you can only see in thick skin slides as opposed to thin skin slides. And it's cells that are in the process of dying. They don't have granules, they don't have organelles, so there's really nothing to stain inside them because all that stuff is degraded. Mostly what you've got here are dying keratinocytes, and internally their keratin molecules are started to reorganize into cable-like bundles to make them flat and particularly resistant. And then, after they're dead, then they just keep on growing towards the surface, stacked on top of one another like very, very flat bricks. Right. Okay, so the outermost stratum of all skin, thick and thin, is your stratum corneum. When you talk about cornified skin, that means that it is dead and packed with keratin. It contains up to 30 layers of dead, scaly, keratinized cells that form the surface layer. So let's show the picture of that one. Here I've got, here we go, the scanning electron microscope shot of the surface of skin with hair follicles sticking out of it. Okay? And it looks like almost like leaves that you raked up in the backyard, but that's what your stratum corneum looks like from the very surface. All of those are your flat keratinized cells flaking from the top. Okay. And of course, because they are flaking all the time, the process called exfoliation, that means that wherever you go, you're leaving a small trail of dead cells. And so where you live is the place where this is going to be falling off the most, which is why most of the dust in your house is made of you. Cells that have flaked off of your hair, flaked off of your skin surface, and now accumulated on every flat surface in your room. Which, of course, brings us to this interesting point. There are things that live off of that stuff. I mean, yeah, they may not be nutritionally powerful, but it's still proteins that other things can feed off of. And this is a close-up view of one of our friends, a dust mite. Dermatophagoides. Derma, having to do with skin. Phago, eat. So it's a skin eater. It doesn't live on your skin. They shouldn't, because they're going after dead cells, and so they are going to be hanging out in the dust of your house and feeding off of all that leftover material. And interesting thing about this, I am allergic to dust mites, but it's not the dust mite itself that causes my allergic reactions, it's dust mite feces. It's their waste that we tend to have allergic reactions to. So, as a dust allergy sufferer, here's some practical pro tips for you. If you wash your bed sheets with regularly, that's a good thing to start. But if you just want to make sure, then put them in the dryer on high heat for an hour. That will not only kill dust mites, but will also kill their eggs. And so that's a good way to make sure that you don't have bad dust allergy reactions if your sheets stay with you for a while. And of course, try to do your best to dust. So yes, we're constantly surrounded by all these little friends. It's a little horrifying. But anyway, getting back to skin. Right. Here's another scanning electron microscope shot showing all the layers of the skin. This part, which is a little bit yellow and looks like, flaky pastry, that's your stratum corneum. Those are the layers of dead cells. And here you can see those layers a little more clearly. And then this sort of grayish area, those are all the living cells. So there's stratum granulosum in here. Most of it is stratum spinosum until you get down here to the place where the colors change. That's where your stratum basale would be. And then all of this is dermis. Dense, irregular, connective tissue with lots of protein fibers that are going every which way all over the place, and here the cells haven't been particularly highlighted, but let me just go back to this image. Right. One like this. Right. So there were the dead cells on the top. That darkest layer right there, that's your stratum granulosum, where you have the cells with the dark staining granules in it, and then this is all stratum spinosum until you get down to the bottommost layer, which is going to be your stratum basale. Right. Okay, so let's see. It's important, I feel, for you to know what a keratinocyte does in order to get from this layer all the way to the top. So, keratinocyte's life cycle goes like this. Let's see. Slide. Basale, dendritic, granulosum, corneum. All right, here we go. Keratinocytes produced deep in the epidermis by cells, stem cells in stratum basale, and they'll just keep on reproducing, making more of them. Right? Some deepest keratinocytes, stratum spinosum, also multiply to increase their numbers. Mitosis requires an abundant supply of oxygen and nutrients, which is why most of the reproduction is happening in the basale closest to the blood vessels. The further away they get, then the slower their metabolism becomes, and eventually they're going to die. Right? Newly formed keratinocytes push the older ones towards the surface. In 30 to 40 days, a keratinocyte makes its way to the skin surface and flakes off. So every skin cell has about a 30 to 40 day lifespan. It gets slower in old age, as does most cellular reproduction, which is why an older person's skin may be more thin and more easily bruised, because there's actually fewer layers of dead cells on the top. If you get calluses or corns, you get thick accumulations of dead keratinocytes on the hands and feet as a result of putting strain on the skin by, say, accumulating a blister first. And the cytoskeleton proliferates as the cells are shoved upwards. So that means that internally, the protein fibers are becoming denser and denser and denser. Now, the cytoskeleton fiber that was made up of keratin were the intermediate filaments. So just imagine that those intermediate filaments are becoming more and more populous inside the cells as being pushed towards the surface. And when you get to the stratum granulosum, you get the lipid-filled membrane-coating vesicles, the granules. Right? Then, let's see, stratum granulosum, 3 important developments. So just before they're about to die, keratinocyte, nucleus, and other organelles degenerate. Cells die. Keratohyalin granules release the protein filaggrin, and that's what's being put outside the cell, binds the keratin filaments together into coarse, tough bundles. Membrane-coated vesicles release the lipid mixture that spreads out over the cell surface and waterproofs it. So, they're tough cells, packed with keratin protein. Outside, they've become a little bit more waterproof thanks to the glycolipids. And there you go. Now you have dead cells that are going to be water-resistant as well as difficult for other things to pass through. So we get our epidermal water barrier formed between stratum granulosum and stratum spinosum. All right. So the dead layer, critical to retaining water in the body and preventing dehydration. Cells above the water barrier die. Dead cells exfoliate, and thus we get dander and dandruff. All right. So let's see how are we doing for time. 12:05. All right. So I'll stop this one, having covered the components of the epidermis. And then we need to get to the structures of the dermis, so that'll be the next video. So see you shortly. Stop.